Good day from Inside Drug Discovery. During this video, we will outline the lead optimization process and describe roles of various types of activity data in the process. From lead selection, we have a lead. Actually, a series of leads, a lead series. During lead optimization, the concept of optimization applies to the properties of the compounds. Which properties? I'll group the 4 properties into 4 areas. Potency, which is the concentration at which the compound exerts its effect. A lower concentration is better and often corresponds to a lower dose for the final drug. Efficacy, the magnitude of the beneficial effect of the compound. ADME and pharmacokinetics, or PK. These are properties that ultimately determine traits like the half-life, absorption, and excretion of a compound. In other words, how a drug gets in a patient, how long it stays, and how it gets out. Finally, safety. Undesirable toxic effects of a compound. These are the key properties. How are these properties tested? Initially through in vitro assays and later with in vivo studies. Let's see how this process works. The process is highly iterative, with feedback in each cycle. Let's say that we start with the lead series and select a new analog. We will perform many tests. These could be multiple in vitro assays or in vivo studies in preclinical species. We would then evaluate the activity data and how the compound performed. We would then consider the results in the context of the available SAR data. This data set would include the activity of other compounds, as well as other information like X-ray data for the target. We would then make yet another new analog, and the process would repeat. By repeatedly going through this feedback cycle, hopefully we can generate a compound that has adequate properties, potency, efficacy, PK, and safety to become a clinical candidate for the discovery program. Here is the same idea shown more visually. The original lead picked from the selected lead series is shown here as a single compound, a big red dot. Modifications to the lead involve making structural changes to the compound to give new analogs of the original lead. The black dots are new analogs. Some analogs will have poorer properties and others will be better. Again, properties is a general idea: potency, efficacy, PK, and safety. Trade-offs are common, as sometimes one specific property may improve at the cost of another. It's messy. One or more analogs may represent notable improvements and be considered a new lead. The preparation of more analogs may afford another improved lead. Eventually, properties may be improved to a degree that the preclinical data support an IND application so that the lead can be advanced into the clinic. That is a quick summary of lead optimization. We have outlined the lead optimization process and described how different types of activity data, efficacy, potency, pharmacokinetics, and safety help drive the preparation of new, more promising compounds. Thank you so much for watching this video. Please subscribe, leave a like, or make a comment. Take care.